Our protocol enables identification of larval neuronal populations, encoding nutritional choices related to the control of the intake levels of two major macronutrients, proteins and carbohydrates. The simplicity and relatively high throughput of our method allows the quantification of food intake for several genotypes exposed to different nutritional conditions.
To genetically cross the parental lines, set up 60 mm embryo collection cages with L3 rearing diet plates supplemented with some active yeast paste. Transfer the adult trip A1 female virgins and Genelia gal4 males, aged 5 to 8 days, to the embryo collection cages, and allow the mating to occur for 24 to 48 hours at 25 degrees Celsius with 60% humidity and a 12-12 light-dark cycle. After the mating period, transfer the mated adult flies to fresh L3 rearing diet plates and allow the egg laying to occur for 3 to 4 hours at 25 degrees Celsius. Make sure that all plates are labeled with the genotype and date of the egg lay. When the egg-laying period is finished, remove the plates from the cages and estimate the number of eggs per plate by counting the number of embryos in 1/4 of the plate. Keep the larval density to a maximum of 200 embryos per plate and cover the plates with plastic lids. Incubate the L3 rearing plates at 18 degrees Celsius, 60% humidity, and a 12-12 light-dark cycle for 9 days. After the